Hello everyone, thank you for being here. My name is Ralf Raumanns. I'm a lecturer/researcher from Fontys University of Applied Sciences and a part-time PhD candidate at Eindhoven University of Technology. I will present our work ENriching Health Data by Annotations of Crowd and Experts. ENhance results from a collaboration between the Fontys University of Applied Science, Eindhoven University of Technology and IT University of Copenhagen. Machine learning offers many opportunities. Machine learning can classify skin cancer with a level of competence comparable to dermatologists. But medical imaging datasets, for example for skin lesion diagnosis, are limited and overfitting can occur. To illustrate this, I included findings from Winkler and colleagues. They found that skin markings in dermoscopic images may impair the diagnostic performance of the convolutional neural network. On this slide, in the top row of the image, left a skin lesion without markings and right the related heat map of the CNN model. The heat map reveals the relevant pixels for the model's prediction of malignancy. As you can see, the model focuses on the nevus, but in the bottom row, the heat map shows that the model focuses on the scale bar. In other words, the model ignores the nevus, it does not generalize well. A promising approach to generalize better is multitask learning, where the model has to learn different tasks simultaneously. This approach showed improved performance in various medical applications, for example for breast lesions. However, when moving from single task to multitask models, we need additional annotations. Typically, the datasets have no additional annotations. To add expert annotations to a dataset is far too expensive and time-consuming. That is why we use non-expert annotations in our research. The non-expert annotations are relatively easy and inexpensive to obtain. For diagnosis of skin cancer, for example melanoma and basal cell carcinoma, a dermatologist uses visual features such as asymmetry, the degree of symmetry of the skin lesion. Melanomas often have an asymmetric shape. Border, the degree of irregularity of the border. Melanomas often have an irregular border. And finally color, the colors present in the lesion. Some colors can indicate melanoma. Co-author Cheplygina reported that ABC scores are good input features even when there is a disagreement between the annotators. It leads to better than random results. We saw that ABC scores are good input features, but how to use the ABC annotations? When offering a new skin lesion to the already trained model, it is practically impossible to ask the annotators for ABC scores. So, we decided only to use the ABC annotations while training the model. Multitask learning facilitates this. The multitask learning model is not only trained with skin lesions and associated labels, abnormal, healthy, but also with the ABC score. So, the model has two outputs with different network heads. One head is the classification output, the other represents the visual feature, such as asymmetry. In our work, we present a dataset of additional annotations for skin lesion diagnosis based on non-expert annotations on three dermoscopic criteria: asymmetry, border and color. The annotations were provided by three different annotation sources. We want to show the possibilities of additional annotations in a multitask modeling environment. The annotations were provided by three different annotation sources: students, crowd and classical image processing algorithms. For annotating, we used two public datasets. The Isaac 2017 Challenge dataset. It was developed for lesion segmentation and lesion classification algorithms. It contains 2000 dermoscopic images with lesions of three classes: 370 melanoma, 1372 nevi and 254 seborrheic keratosis lesions. The second public dataset we used was the PH2 dataset, which was acquired at the hospital in Portugal. It contains 200 demoscopic images, of which 80 common nevi, 80 atypical nevi and 40 melanoma lesions. The dataset also contains expert annotations of lesion characteristics, including color and asymmetry. In the next slides we zoom into the different annotation sources. We start with student annotations. Co-author Cheplygina gathered the annotations by students in an educational context. The students learned about medical imaging and data annotation and were encouraged to decide on their annotation protocol based on existing literature. Each group could decide which visual features they wanted to focus on and the type of annotation scale they wanted to use. They annotated visual features of 100 images. Each visual feature was annotated by at least 3 students from the group. Students were not blinded to the diagnostic label while annotating. Since this is a public dataset, anybody could try to find the label. Only the Isaac skin lesions were part of the student assignment, as the PH2 dataset was added to this after the subject was no longer taught. The crowd is the second source we use for additional annotations. Crowd workers on Amazon Mechanical Turk received instructions on rating the ABC features based on 3 examples. The images on this slide illustrate how the annotation task is presented to crowd workers. The annotation scales were as follows. For asymmetry, a 0 for a symmetrical lesion, a 1 score of half symmetrical lesions, and a score of 2 for asymmetrical lesions. For border, the crowd scored the degree of irregularities in the skin lesion's edge with a maximum score of 8. A score of 8 indicates irregularities in the entire edge, a score of 4 in half the edge, and a score of 0 indicates no irregularities. For color, the crowd counted the number of the colors being present in the lesion. Only light brown, dark brown, white, blue-gray, black and red had to be considered. We requested 3 different crowd workers to annotate each skin lesion. The diagnostic labels were not provided, but due to the public nature of the dataset, could potentially be known. But this is less likely for MTurk because MTurk jobs are time limited. Only workers who had previously done more than 500 approved tasks with an approval rate higher than 90% could participate. We paid the workers $5 per task. The workers annotated 1,250 lesions for Isaac and all 200 lesions for PH2 dataset with asymmetry, border and color annotations. Finally, classical image processing algorithms are the third source we use for additional annotations. We used classical image processing algorithms from the literature to automatically extract asymmetry, border and color features. The image on this slide shows the different calculation steps that the 3 algorithms take. The algorithms use binary masks as input. The color score algorithm also uses the skin lesion image itself. On this slide, a summary of the collected annotations. Let's start with student annotations. Stud A stands for student asymmetry annotations, Stud B for student border annotations, and Stud C for student color annotations. All groups annotated at least some of the ABC features, resulting in 1,631 images with asymmetry, border, and color annotations. Crowd annotations. Crowd A, Crowd B, and Crowd C for color. The crowd workers annotated 1,250 lesions from Isaac and annotated 200 lesions from the PH2 dataset with asymmetry, border, and color annotations. The classical image processing algorithms had the automated annotations, Auto A, Auto B, and Auto C, annotated 1,970 asymmetry annotations, 1,996 border, and 2,000 color annotations for the ENriching dataset. For the PH2 dataset, 165 symmetry annotations and 200 color annotations were collected. The expert annotations were already present in the PH2 dataset, so 200 for asymmetry and 200 for color. Let's zoom into annotation analysis. Is there a correlation between the annotations and the diagnostic label? What is the level of agreement between annotation sources for the asymmetry, border and color criteria? To gain insight into the characteristics of the collected annotations, we use visual representations that provide detail and overview at the same time. In particular, we created rain cloud plots with A, B and C annotations for each annotation source. The plots show the distributions of asymmetry, border and color scores versus the label, normal or abnormal, for each annotation source, where diagnostic label 1 stands for abnormal and diagnostic label 0 stands for healthy lesions. We normalized the scores per annotator, resulting in a standardized data with a mean of 0 and standard deviation 1, and then averaged these standardized measurements per lesion. The first insight that we gained was that the distributions of scores for each diagnostic label overlap considerably. Furthermore, for stud A, stud B, stud C, crowd A and crowd C, the sample medians for abnormal lesions have a higher value than the sample medians for normal lesions. So what about the PH2 dataset? What we can see is that the annotations of the expert asymmetry are right skewed for normal lesions and left skewed for abnormal ones. Auto-A's distributions for abnormal and normal lesions overlap considerably, and the sample median for abnormal is higher than the sample median for normal ones. Besides rain cloud plots, we also used the Pearson's coefficient measures between annotation type and the label. The measure indicates there is no correlation between the crowd B, Auto-A, Auto-B annotations and diagnostic label, but crowd A, student B, automated C, crowd C and student C have discriminative power. The second research question examined the level of agreement between annotation sources for the A, B and C criteria. In order to answer this question, the results of the analysis of agreement level using Pearson correlations were examined. For asymmetry, we see a moderate positive correlation between student and crowd annotations. For color, we see a weak correlation between all annotation sources. For border, there is no correlation between the different sources. Based on these agreement levels, annotating the asymmetry and color seems more straightforward than annotating the border. Let's continue with the experiments. To investigate the added value of our annotations, we address a binary classification problem, healthy, nevi, versus abnormal, melanoma and seborrheic keratosis. We performed multitask learning with the help of 3 different architectures, VGC16, Inception and ResNet. So why these 3 architectures? In 2017, during the Skin Lesion Challenge, Deep Residual Networks, ResNets, performed melanoma detection and achieved state-of-the-art performance. Furthermore, the VGC16 and Inception models have demonstrated effectiveness in computer vision tasks. All 3 networks have great potential for our case study. To investigate the added value of our annotations, we utilized these 3 different architectures as a baseline and the multitask model. To understand how the collected annotations can contribute to training deep learning algorithms, we performed several experiments where we compared a baseline model which is trained only on the diagnosis, a multitask learning model which is trained on both the diagnosis and one or more of the collected Isaac-based ABC annotations, an ensemble of multitask learning models, and finally a multitask learning model with synthetic annotations. To investigate the value of the additional annotations, we compare baseline networks with multitask networks, to which we add one of the following 9 types of annotations individually. For asymmetry, Auto-A, Crowd A, Stud A. For border, auto B, crowd B, stud B, for color, auto C, crowd C and stud C. We also combined the networks above into ensembles by averaging the network predictions with equal weights. We tested the following three ensembles. A combination of stud A, stud B, stud C. The ensemble is named stud ABC, a second ensemble named crowd ABC and a third ensemble named auto EBC. Finally, we trained the ResNet 50 multitask learning models with synthetic annotations. So what we did is we replaced the original A, B and C scores with uniformly randomized ones in the range of 0 and 1 and combined them into ensembles. Our experimental setup. We trained all CNN models using 70% as training, 17.5% as validation and 12.5% as test proportions in a 5-fold cross-validation approach. So we used 5-fold cross-validation with the data split in a stratified fashion, keeping the malignant-benign ratio equal over the training, validation and test subset. We trained all layers, including the convolutional base, starting with the pre-trained ImageNet weights. For training, we used 30 epochs with a batch size of 20, using the default backpropagation algorithm, RMS-prop, as the optimizer, with a learning rate of 2 × 10⁻⁵. We did not freeze the convolutional base to train the model as best as possible. In other words, we continuously updated the weights of the convolutional base. The multitask learning networks are combined into ensembles by averaging the network predictions with equal weights. Earlier experiments showed limited effectiveness of optimizing the weights. So, what is the effect on CNN performance for multiple annotation usage? We compared the average area under the curve of the baseline model to the average area under the curve scores for the different multitask models and ensembles. The average area under the curve score was calculated per experiment, taking the average of the AUC score of each fold. So some insights. The AUC values for each CNN architecture are close together. The Inception and ResNet outperformed the VGC16 architectures for all three features. The three ensembles show improved performance when compared to the other models in the same architecture. The best performing model is the Auto ABC ResNet 50 ensemble model. For VGC16 and ResNet, most multitask models outperform the baseline model. If the annotations were perfect copies of the diagnostic label, this would not add information to the classifier, so some disagreement is necessary. To emphasize the point, we performed an experiment where we used randomly generated annotations instead of our annotations. We replaced original A, B and C scores with uniformly randomized ones in the range of 0 to 1 and combined them into ensembles. We compared synthetic annotations to original annotations for ResNet 50 ensembles. The table shows that ResNet 50 ensembles trained with retrieved annotations perform better than ensembles with randomized annotations. When compared to the baseline, there is still some generalization when synthetic annotations are used, although less effectively than our annotations. One perhaps surprising result is that noisy annotations positively contribute to the CNN performance. Our annotations are highly noisy, as shown by the rain cloud plots and the correlation coefficients. We explain the success of these annotations by several factors. First, annotations that are perfectly correlated with the diagnostic label would not add information to the classifier. Second, training multiple classifiers and combining these in an ensemble is a well-known result that can lead to improved generalization. We also showed that this is the case for fully randomized annotations, but that this is less effective than our gathered annotations. Let's continue with our summary. We see at most moderate correlations between annotations and the diagnostic label. Furthermore, we see weak to moderate inter-agreement level between the different annotation sources. Finally, we demonstrated the added value of using non-expert and automated annotations for model learning. What are the next research directions? We will continue to focus on bias and multitask learning in the coming period. So, to what extent are we sure that datasets represent the population they are applied to? To what extent are we sure that models and algorithms represent the population they are applied to? Bias is a very interesting topic that deserves more attention. One thing we are going to do is transfer the knowledge of our case study to another domain, for example to lung nodule diagnosis. However, there are still many interesting research directions, for example finding methods to discover and avoid shortcuts. Is it helpful to skip a skin lesion during training when there is low agreement on the ABC score between the different annotators? What is the trade-off between quality and quantity of annotations? Multitask learning prediction of ABC scores with diagnosis helps to explain the diagnostic outcome. What kind of model architectures fit the best for this explanation task? What types of annotations are suitable for this task? And is it possible to take into account the level of expertise of the annotators? We hope that our dataset can be used in and inspires further research into multiple annotations and/or multitask learning. All data and models are available on GitHub. We want to thank the MELBA journal and the peer reviewers. Thank you for your attention. If you have any questions or comments you would like to send to me, my email address is ralf.raumanns@fontes.nl. All the best!